Hello students, today we are going to learn about certain creatures which were not given any kingdom by Whittaker in his 5 kingdom classification. They are the viruses. Whittaker did not assign any kingdom to viruses although they are so abundantly found in nature. The question is why? Why didn't he assign any kingdom to viruses? Because Whittaker had assigned these kingdoms to living organisms. Who is called a living organism? The criteria for a thing to be called as a living organism, it needs to have minimum one cell, which the bacteria had, the protists had. That's why they were given kingdoms. But virus, what is virus? It's a speck of protein enclosing a genetic material inside. That's all that it is. Okay, if a bacteria is so big, a virus will be like a small speck inside it. So it did not have a cell. So how can it be called as living organism? It could not be called as a living organism, and that's why it was out. It was not assigned any kingdom. We are learning about viruses. They are said to be acellular. What does this mean? Without a cell. And that is the reason why they are not truly living, and therefore they were not assigned kingdom in Whittaker's 5 kingdom classification. So if they are not living organisms, then what are they? Are they non-living? Can they be considered as non-living? Outside a living cell, they are just like a speck of dust. So inert, it won't be able to do anything. It will just be carried passively to different places. Its activity begins only when it is inside a cell. That is when it will take over that cell's machinery. It will hijack that cell as its own and make that cell produce its own proteins, replicate its genetic material, do activities for itself. It will hijack the host cell's machinery. That is when it is inside a cell. But when it is outside a cell, it is just like a speck of dust. Inert crystalline particle is what it is called outside a cell, also called as virion. Let's write this down. Have an inert crystalline structure. Inert means what? It will not be able to react to anything outside a living cell, and here they are called as virions. Once inside a host cell, they will take over the host cell machinery. Important sentence from NCERT. Why do they take up the host cell machinery? In order to replicate itself. So they are not called as living organisms. So are they non-living? No, they cannot be called as non-living also, because they are capable of doing things that only a living organism can do. For example, they can cause diseases in the host cell. They are so host specific. Okay, they will not enter into any cell. They will enter into that particular host cell only. That's called host specificity. Causing infection. These are the characteristics of it being living. What are the non-living characters that it shows? Being very inert, absolutely inert. There is no growth, there is no metabolism, no replication outside a cell. That means that it has characteristics of both living as well as non-living. It lies on the borderline of living and non-living. You have learned this statement, right? In your previous classes. Have characteristics of both living and non-living. What are the living characteristics? Cause infections, diseases, host specificity. Non-living features include features like no growth, no metabolism, no replication, etc. They are said to be obligate intracellular parasites. Obligate means compulsory, intra means inside cell parasites. To show any characteristic feature of a living, they have to compulsorily be a parasite inside a living cell. Obligate intracellular parasites. Virus actually means venom or poisonous fluid. They are so small that they are almost fluid. So we are looking at the discovery of viruses now. Discovery of viruses. So it was the year 1892. Tobacco plants were suffering from a disease wherein the leaf lamina was getting such kind of yellow kind of patches. These are called as mosaic, and since the tobacco plant was having this disease, it was called as the tobacco mosaic disease. Viruses were not discovered till this time. Only bacteria were discovered, so everybody assumed that probably it is some bacteria that is causing this disease, or it could be some condition with the plant only, internal condition. So came the scientist Dmitry Ivanovsky, took these leaf samples, he crushed them into a pulp, made a slurry, made a fluid out of this. In order to separate the bacteria from this fluid, he passed this fluid through bacteria-proof filters. It was expected that the bacteria inside this fluid would be trapped onto the filter so that he would be able to isolate the bacteria. But go to see, no bacteria were captured on this filter. Not only that, this fluid could cause infection in healthy plants also. What did it mean? That this infectious agent is smaller than the size of a bacteria, and maybe it's something in the fluid itself, because this fluid is highly infectious. It can cause diseases in healthy plants. He found that the causal organism for tobacco mosaic disease is smaller than bacteria, as it could pass through bacteria-proof filters. Next, in the year 1898, few years later, the next scientist, M.W. Beijerinck, he repeated Dimitriev's experiments, and he stated that the causal organism is inside this fluid smaller than a bacteria. This infectious agent is this fluid only, because he was not able to isolate any organism from this. He said that it cannot be a living organism, it's this fluid itself that is infectious. He stated that the extract from the infected plant could cause diseases in healthy plants. He stated that this fluid itself is infectious. He called it as contagium vivum fluidum. He stated that extract from infected plants could cause disease in healthy plants. He called the extract as contagium vivum fluidum, meaning infectious living fluid. Then came third scientist in the year 1935, W.M. Stanley. He purified this fluid and he evaporated it, just like how sugar solution is evaporated. What is left behind? Crystals of sugar are left behind, isn't it? Just like that, he evaporated the fluid, and what he found was crystals were left behind, which were mainly made up of proteins, which represented the virus, because viruses are mainly comprised of proteins. So he said it was an important point for deducing the structure of the virus. He said that the viruses could be crystallized, and the crystals were mainly made of protein. Important sentence from NCERT: viruses could be crystallized and the crystals were mainly made up of protein. So these were the key features in the discovery of viruses. Next we do the structure of viruses.
महादेव के नाम पे सब्सक्राइब कर दें।